Everyone, today we are here to discuss the previous year NEET questions on body fluids and circulation. This is the part 1. So moving on to the first question. Adult humans RBCs are enucleated. Which of the following statement is our most appropriate explanation for this feature? Options given are: they do not reproduce, they are somatic cells, they do not metabolize, all the internal space is available for oxygen transport. So let's look at the options. So they do not reproduce. Yes, this is true, they do not reproduce. But is that why they do not have a nucleus? No, it's not because of that. They are somatic cells. Yes, they are somatic cells, they belong to the body. They do not metabolize on their own. So yes, they do not have their own mitochondria, which is the powerhouse of the cell, so they do not metabolize on their own, but they only use anaerobic metabolization. So but the fourth option, all the internal space is available for oxygen transport. Yes, let's look at the structure of RBC. So this is the RBC. So there's a small depression here. So when we look from the side, it looks something like this. There's a depression on both sides. Since there is no nucleus inside the cell, the size, this looks something like a dumbbell. So here the oxygen can sit with the hemoglobin. So there can be oxyhemoglobin or carboxyhemoglobin for the transportation. So that is the reason why matured RBCs are enucleated. So the answer is only 4. In human RBCs, nucleus gets degenerated during maturation and it provides more space for oxygen carrying pigment hemoglobin. It lacks many cell organelles including mitochondria, so they respire anaerobically. So the next question is: reduction of pH in blood will option A reduce the rate of the heartbeat, option B reduce the blood supply of the brain, option C decrease the affinity of hemoglobin with the oxygen, option D release bicarbonate ions by the liver. So let's look at a small chart that we know already. This is the oxygen hemoglobin dissociation curve. So this is sigmoid shaped, we know, because the saturation of hemoglobin with the concentration of oxygen varies with regards with the concentration. It is not constant. If it is constant, it would have been linear. So if we shift the graph towards the right side, it means that the HbO2 is decreasing. But when the graph moves to the left side, I'm sorry, this is the right side, this is the left side, the HbO2 increases, oxygen binding increases. So for this to occur, we have to remember only one thing: low pH and low CO2. So rest all should be high. We can have high temperature or higher CO2 for more dissociation of oxygen into hemoglobin and oxygen. So HbO2 will get reduced. So only low pH and low CO2. Rest all are high temperature, H plus ion concentration are high. So this is called acidosis. This is called alkalosis. Okay, so acidosis leads to decrease in the affinity of hemoglobin with the oxygen. So the option is C. Decrease the affinity of hemoglobin with the oxygen. Reduction of pH of blood will decrease the affinity of hemoglobin with oxygen, which in turn causes acidosis. Which one of the following is correct? Serum is equal to blood plus fibrinogen. Option B, lymph is equal to plasma plus RBC plus WBC. Option C, blood is equal to plasma plus RBC plus WBC. Option D, plasma is equal to blood minus lymphocytes. So serum is equal to nothing but plasma minus fibrinogen. So when we have let the blood to clot, and there is a fibrin that formed here in the plasma region. So in a test tube, when all the cellular components have settled, and here is the plasma. So after clotting has occurred, the fibrinogen will also go out. So when the plasma is devoid of fibrinogen, it is called serum. What is lymph? When the blood flows through capillaries, some amount of fluid and WBC escape out. Fluid and WBC. So this again gets back into the lymph nodes, drained into the lymph nodes. So then this becomes lymph. So this is extracellular fluid, does not contain any RBC or proteins. So blood is equal to plasma plus RBC plus WBC. This is correct. Plasma is equal to blood minus lymphocytes. This is also wrong. Plasma is equal to blood minus cellular components as a whole. So blood consists of plasma, RBC, WBC and platelets also. So the solution is option C. Erythropoiesis starts in option A, liver, spleen, red bone marrow, kidney. So we have to see which are all the erythropoietic organs. So erythropoiesis occurs in children, in embryos from about in the first month. In the first month it occurs in yolk sac. Yolk sac is a embryological structure. Only temporarily it produces, only for few days. And then after 5 weeks, after 5 to 6 weeks, not months, weeks, it is followed by liver. And then after 6 months, it's by a red bone marrow only. Throughout the lifetime, it is only by red marrow. So primarily, it is only red bone marrow we have to consider it as erythropoietic organ. So the answer is option C. Erythropoiesis is the process of formation of RBC. In the first month of pregnancy, yolk sac is the hematopoietic tissue. After 5 weeks, it is followed by the liver. Red bone marrow from the 6 months onwards becomes the principal site of erythropoiesis. So the option is C. Person with blood group AB is considered as universal recipient because he has options given are both A and B antigen on RBCs but no antibodies in the plasma, both A and B antibodies in the plasma, no antigen on the RBC and no antibody on the plasma, both A and B antigens in the plasma but no antibodies. So AB blood group person, both A and B antigens are present on the body but there are no antibodies at all in the plasma. So we have to remember this. Only antigens are present but no antibodies. So let's read the explanation. Carl Landsteiner was the first person to give the ABO grouping. This can also be a question. So there were A, B and O blood groups. So Castillo and Staney found the AB blood group. They found the ABO grouping but they found the AB blood group. ABO is determined by the gene I, agglutinin. There are 3 alleles IA, IB and IO of this gene or small I. The protein produced by the IA and IB alleles are called A antigen and B antigen, that is on the surface. Individuals with the AB blood group have both antigen A and B on their surface but no antibodies for either of the antigens. That is why when there is no antibody they can accept blood from anyone. So that is the reason why they are called universal recipients. So moving on to the next question. The figure shows a human blood cell. Identify its given characteristics. So this is a cell with small large number of granules, no particular shape or size. So this can be basophil. This is basophil. So what does basophil do? It is involved in inflammatory reactions. It produces serotonin and heparin and major histamine. Okay, heparin is anticoagulant. So basophil. So the answer is basophil secrete serotonin inflammatory response. Monocyte is larger in size. It does phagocytosis. B lymphocyte is also lymphocyte larger in size. Neutrophil is also WBC that is involved in granulocyte that is involved in phagocytosis and inflammatory responses. So according to the morphology, it's only basophil. Basophil secrete histamine, serotonin, heparin are involved in inflammatory reactions. So the next question is, which one of the following plasma proteins is involved in coagulation of blood? So the options given are an albumin, serum amylase, a globulin, fibrinogen. So albumin is produced by liver. It maintains the colloidal osmotic pressure. What does serum amylase do? In our previous videos of digestion absorption, we have talked about the serum amylase, which acts on starch. So it does not do anything to the blood. What is globulin? Globulin is involved in defense mechanisms, alpha, beta, gamma globulins. Okay. So only fibrinogen produced by liver is involved in coagulation. Fibrinogen occurs as fibrinogen, which is inactive form. It converts the fibrin in presence of thrombin. Okay. So fibrinogen is majorly required for coagulation of blood. Fibrinogen, the protein of blood plasma, is converted into insoluble protein fibrin during the clotting process. The fibrinogen free fluid obtained after removal of the clot called blood serum So that is what serum is, plasma minus fibrinogen. So the next question, so the answer is a fibrinogen. Which one of the following organs is called graveyard of RBC? Gallbladder, as we know, is more important in digestion. We have seen in our previous video, digestion. It stores and concentrates the bile produced by the liver. And kidney is majorly involved in excretion. It also produces renin, that is more involved in the vasoconstriction process and RBC reduction process, but not the graveyard of RBC. Liver produces many proteins, clotting factors also, fibrin, albumin, most of the proteins, but it does not destroy RBC. Spleen is the graveyard of RBC, where it destroys the RBC and takes back the hemoglobin. We know the lifespan of RBC is only 120 days, and then it reaches the spleen so that the hemoglobin can be extracted and can be used again for the production of new RBCs. Spleen is an organ of lymphatic system located in the left upside abdominal cavity under the diaphragm. So it is under the diaphragm. We have the stomach here, and we have the small intestine, pancreas. Here we have the liver, gallbladder, and here we have the spleen. Spleen is under the diaphragm. It is called graveyard of RBC because fragment of RBC cells, old and dead cells, are constantly being removed from the bloodstream by it. So the answer is spleen. ABO blood groups in humans are controlled by gene I. It has 3 alleles, IA, IB and I, small I. Since these are different alleles, 6 different genotypes are possible. How many phenotypes are possible? So we have IA, IB, I. So these are alleles. So there can be genotype and phenotype. What is genotype? Genotype is the gene type. That is the gene expression. How is the gene pattern? What is phenotype? The generalized external appearance, or how it comes out as a whole process. So we can cross match and see. With IA, we'll have IA, IB, IA, I, and with IA itself we have IA, and we'll take IB here. This is for IA. We'll match IB with IA. So with IA, IB, it will become IA, IB, it will become IB, and with IB small I, it will become IB small I. And small I, it will become I, and small I, IB, and small I. So IB. So this will become AB group. So IA, this will become A group. IB and BI, this both will become B group. These three will become B group. So II will become O group and IA will become A group. So what we have to remember majorly is IA, let's take IA, IB and small I or IO. So IA is recessive. It is recessive to both IA and IO. So IA, I will mean it's only A group. Genotype is different, but it's only A group. For B also it is the same. But IA and IB cannot dominate over each other. When IA, IB come together, it is only AB blood group. So the 3 alleles in ABO blood groups in human can produce 6 different genotypes or 4 different phenotypes. So genotypes according to the blood group A are IA small I. Blood group B are IB small I. Blood group AB are IA and IB only. Blood group O is only possible in II. So the answer is 4 phenotypes. What are true about RBCs in human? They carry about 20 to 25% of the oxygen. They transport 99.5% of the oxygen. They transport about 80% of the oxygen only, and the rest 20% of it is transported in dissolved state in the blood plasma. They do not carry CO2 at all. So reading the first statement, they carry about 20 to 25% of the CO2. That is actually correct, as carboxyhemoglobin or carbaminoglobin. Okay. So in B, they transport 99.5% of oxygen. So oxygen is only 97% exactly. The other 3% is by dissolved forms. They transport 80% of oxygen. No, it is again 97. And 20% of it. No, it's only 3% here. They do not carry CO2 at all. No, this is not possible. They carry CO2. So the answer is option A. They carry about 20 to 25% of CO2. Blood transports oxygen from respiratory organ to the tissue cells and also transports carbon dioxide from tissue cells to the respiratory membrane. About 97% of the oxygen is transported by RBC in the blood, while remaining 3% of the oxygen is carried in the dissolved state through plasma. Nearly 20 to 25% of carbon dioxide is transported by RBCs, whereas 70% is carried as bicarbonate. Another 7% of CO2 is carried through dissolved form. 70% is through bicarbonate, HCO3 minus ions. So the answer is given. The most popularly known blood grouping is the ABO grouping. It is named ABO and not ABC because O refers to O or 0. So we can, for easy remembrance, we'll say 0. Then we'll understand why. Over dominance of this type of genes for A and B types. One antibody only, either anti-A, anti-B on RBC. No antigens A and B on RBC. Other antigens beside A, B on RBC. So the ABO blood grouping system is most important blood group system in human blood transfusion. It is named ABO, not ABC, because the blood group O or 0 in some countries, individuals do not have A or B antigen. They do not have A or B antigen at all on their surface, but both anti-A and anti-B antibodies against the AB blood groups are present in the serum. Therefore, the group O can receive blood only from group O, but it can donate to any of the individuals. Okay, that is the reason why we have named it O. No antigen A and B on RBCs. Compared to blood, or lymph has. So, what is lymph? Lymph is the ultrafiltrate of the blood. So when fluid moves out, some amount of WBC can go out through diapedesis. Not diapedesis exactly, but can filtrate through the pores and move. But RBCs cannot move like that. So the lymph will have some amount of WBC. That's why lymph is rich in WBC. So it contains more WBCs and no RBCs. So lymph is mobile connective tissue comprising lymph plasma and lymph corpuscles. Its composition is like blood except that it lacks RBC and large plasma proteins. So the option is option B, more WBCs and no RBCs at all. There is no DNA in option A, matured RBCs, option B, matured spermatozoon, option C, hair root, option D, enucleated ovum. So DNA is basically present in nucleus. So here the indirect question is which does not contain nucleus. So as we know by NCERT fact that mature RBCs do not have nucleus, so that they can accommodate the hemoglobin here. So the option we have to choose is RBC. As with spermatozoon, they do have nucleus. Hair root, hair is basically a non-living tissue. An enucleated ovum does not have RBC, but with NCERT facts we have to choose mature RBCs only. Red blood cells are most important type of blood cell and delivering oxygen to the blood tissues via the blood. There is no DNA in mature RBC. The reticulocyte is the immediate precursor of the mature RBC. Within 24 hours of release into the peripheral action, evolve into mature tissue. So the reticulocyte is the immediate precursor. Reticulocyte is generally larger in size and contains nucleus. But when we are about to get into matured RBC, it will eventually disappear. Nucleus will disappear. So the option is matured RBC. Globulins contained in human blood plasma as primary involved in. So the osmotic balance is kept by salts like sodium, potassium, etc. Oxygen transport is done by RBCs and hemoglobin. The clotting of blood is done by clotting factors and fibrinogen. So globulins are basically involved in defense mechanism. Globulins can be alpha, beta, gamma. Okay. So defense mechanism is by globulins. Globulins contained in human blood are primarily involved in defense mechanism of the body. Globulin is one of the two types of serum proteins, the other being albumin. Globulins can be divided into three fractions based on electrophoretic mobility. Most of the alpha and beta globulins are synthesized by the liver, whereas gamma is produced by lymphocytes and plasma cells in the lymphoid tissues. So the answer is defense mechanism of the body. Which type of white blood cells are concerned with release of histamine and natural anticoagulant heparin? So as we saw before, histamine, serotonin and heparin are produced by basophils. So the answer is basophils. Neutrophils are they are basically non-inflammatory reactions. Eosinophils produce histamine also, but mostly in inflammatory reactions. Heparin is very unique to basophils. Monocytes are involved in phagocytosis. Basophils is type of WBC that has lobed nucleus surrounded by granular cytoplasm. Basophils are produced continually by stem cells in the red bone marrow and move about in amoeboid fashion. This is their movement type. Like mast cells, they produce histamine and heparin as part of body's defense site of action. So mast cells also produce histamine and heparin, an additional fact to be known. So the answer is basophils. Thank you for watching the video.